Great. Our next speaker this morning is Tom Yankee. Is that pronounced right, Tom?
Yes.
Perfect. Thank you. Tom's with the Lower Weezer River Weed Management Cooperative, and he's been a member of that organization since 2007. His previous career was with the NRCS, and Tom has served in several locations throughout Idaho. And he retired in 2007 after 33 years of service. And Tom's talk for us this morning is weed suppressing bacteria from the outlook of this cooperative. Tom.
Thank you for that introduction. I'm really kind of out of my element here. I'm not used to doing these kind of things anymore. And so whenever I'm in a room full of scientists, I think, oh boy, really out of my element. But whenever I hear a lot of you folks speak, I get really excited about things. And 2 years ago, I was at this conference, and Dr. Ann Kennedy made a presentation that kind of fit right in with some of the things we were attempting to do with our cooperative weed management area and some weed problems we had. So the majority of the data that we have here that I'll show you today is actually her information. We've spent quite a bit of time with her in the last year working on different things and different problems that we've had. So if you were coming to this expecting to hear her, unfortunately you get me, because she uses lots of props and she's really interesting to listen to in a talk. So so our mission as a cooperative weed management area is to, we function as an advisory capacity to our county commissioners, and weed control is our focus for controlling noxious weeds, utilizing available programs and partners. And the other thing that should be added to that also is in methods. When you think about weed departments in various counties, you probably think about how many drums of chemicals they may use in a year and these kind of things. But and sometimes you have to, and we also do that in ours, but we also use some other techniques that hopefully aren't quite as invasive, problematic. And here's our fine group. I was thinking about this in diversity of things. So in our group we have a row crop farmer, we have a high school biology teacher, we have livestock producers, and we have hay and forage producers, and we have an individual that manages a ranch for an outside party. So we're kind of a little diverse group of our own. One thing we've been doing for the past 11 years is goat grazing for leafy spurge control on the Weezer River corridor from east of Weezer up through Cambridge, if you're familiar with that area. One thing that happened with this, and by doing it every year, what we've had in the last 11 years is 75% reduction in the amount of days that the herd is grazing, and a 70% reduction in the amount of leafy spurge. This particular area is fairly inaccessible and hard to get to, so chemical application is not an option, but our goat grazing project is working really well. It's made a huge impact in that area. And also our groups collected AppFolio beetles in Montana and transported them and moved them, and using them in combination has also worked very well in a lot of areas. Really how we got into our project was our group had for a number of years had a long discussion about invasion of jointed goat grass in our area. Jointed goat grass along with the other two, Medusa and Cheatgrass. We've got lots of those other two, but we were trying to figure out a way to kind of get a handle on stopping jointed goat grass if we could. So we, as an ex-agency person, what's the first thing you do? Well, you better do an inventory so you know what you've got. So we come up with some funding from some various agencies and those kind of things to do our inventory with. Have a lot of partners, and we also geared this because we have livestock producers in our group towards what may happen with listing of sage grouse. And we felt like the goat grass was going to be an invader, be the next worst thing after Medusa. So we developed a scenario of inventory that predominantly geared to our rangeland habitat areas. We had approximately 300 to 400 miles of roads in those areas, and we inventoried all those roads for density of jointed goat grass. We already knew we had cheatgrass in those areas and that. And so what did we find out? Well, we found out that we had jointed goat grass on 300 to 400 miles of road. So what do you do next? It's like, we kind of had an idea that was going to happen, but it's like, when it gets you, it's just like, okay, now what? This is where the information from Dr. Kennedy actually kind of comes into play, because in a lot of places we may not want to be using herbicides on jointed goat grass because of some native plants or other species, and it's really not cost effective to do a lot of that herbicide work. So to kind of continue on here, we know that they're very invasive exotics. They germinate in the fall and spring, and very competitive in low temperature with root growth. They have lots of tillers and seed and compensatory or plastic, and I don't know if any of you are not familiar with jointed goat grass, but it's the one here at the bottom. It's a relative in the wheat family, so it can be a real problem in cropland areas as well. So with weed suppressive bacteria, the ones, a lot of the isolates that Dr. Kennedy uses, they colonize residue and roots. They survive well at low temperatures, not competitive at warm temperatures, and they're kind of dormant in the summer, and they produce and deliver weed suppressive compounds to the weed root, and they do not suppress crops or native plants. And it was quite a process that her ARS group went through to come up with a lot of these different isolates. So based on the other information that biocontrol could be a good match where you have your soil bacteria that will work and inhibit root growth. And the picture over here, we've got cheatgrass in winter wheat, and with the combination of using a soil-borne bacteria and a plant that's not susceptible to the bacteria, you could have some competition effect that will actually reduce the density of the cheatgrass. Now this one's a little hard to see. This is the control in the plot. You can see the inner space here has lots of cheatgrass in it. And over here where the bacteria has been added, the inner space is relatively clean. There's still some in it, but not a lot. So that makes it a good match. Okay, so there's the control again. And here's with the bacteria. So the isolates that Dr. Kennedy has been working with do not inhibit the growth of and vigor of our broadleaves or dicots. Does not inhibit grasses, monocots, cereals, being cereal grains, native and introduced grasses, or economically important grasses. And you may wonder, okay, so what's an economically important grass? Well, that could be turf grasses and those kind of things. It may be golf courses, those kind of areas. And also, they've been proven through her research that they are ecologically safe because they are native in the environment to begin with. They grow at low temperatures. The genes are on the chromosomes in multiple places, and if you asked me to explain that, I would say I couldn't. You better talk to the previous speaker because he could tell you better than I how all that works. But up here, this little figure is the chromosome. And as much as I know about it, the little dots in there are the plasmids, but all that stuff's way beyond me. So there's no antimicrobacterial activity and no enzymes that, and I don't know if this is the right pronunciation, that lyse cells. Okay, so I had to ask her, what does that term mean? And she says that the bacteria does not puncture or enter the cell of the plant root. And hopefully that's correct. Because if it was to enter the cell of the other plant, then there's a lot of other things that could go on that could change all the chromosomes, and a good thing might turn into a bad thing. And it has no inhibition on Daphne and Lemna. So there's also been toxicology studies that have been completed. It's found that it do not inhibit the Daphnia, the Lemna, or bees, or fish, or birds, or in this case, mice. That's really all of this is very important when you think about the future aspect of applying a product like this on wide-scale plantings for existing scenarios of plants. This chart kind of gives you an indication of reduction. These plots that she had out were in central and southern eastern Washington. So we have cheatgrass plants on this axis, and then years of application on the bottom. The thing about the isolates that she uses is that, with a herbicide, you kind of have an instant response or a fairly rapid response. This kind of a situation is different than that because it takes a while for the population to grow and inhabit the roots. So you can see at approximately here, we're at year 1. There's only a 20% reduction approximately in the plant percentages in the plots. But as time goes on, and if you have beneficial plants in the situation, whether it's a seeded grass species or a native grass species, or it's in cases of cereal grains for competition, as those species increase, gain more vigor, then your reductions become greater. So here by year 3, for a lot of these, we're down to less than 40% of the population, and by year 5, in all these plots, basically we're down to 0. So over time, we need the competition to help reduce the weed impact. So when does it not work? Obviously the bacteria must survive. It does not survive on soil surface because of UV light impacts on it. It needs to be applied in fall when we have soil moisture, which is in less than 50 degrees temperature. That can be a real problem. In a lot of our dry climates, we don't have fall moisture, and we need to have some type of a delivery system involved, how to get it onto the area. The bacteria must establish and needs to be able to overwinter. There may be some problems with some clay soils that may find the toxin, and it inhibits the weed, but plant competition will sustain weed suppression. So an application of the various isolates would be 1 pint of active culture per acre. That would be mixed in a tank mix of probably 20 to 30 gallons of water per acre. Log 8 cells per square meter. I was going to take that off of this slide. I didn't get that done because all I know about log 8 is that it means that there's billions of them per square meter. So we can do a land apply with the ground rig, or it could be applied aerially. It can be coated on seed prior to planting. or in pellets, in pellet form. Pellet form could be really useful in a lot of different applications, particularly with aerial application in late fall and winter. It mostly compatible with most herbicides except for a few adjuvants, and cost per acre estimated to be at the low end of a herbicide cost. That is yet to be determined because it's not in production large enough to be applied on large acreages. This kind of gives you a little idea of reduction based on some of the bacteria. These are some of the different isolates. You can see that applied over different years, that some of the isolates have different effects between years. Some of that could be based on moisture, climate, those kind of things. Some of them don't hardly have any effect. So there's a huge amount of them, of different isolates, but only a few of them are effective on the ones we're really attempting to use. So they varied in their survival and ability to inhibit the weeds in the field. So we're partially at a conclusion here, but so what we know is soil is alive and has all sorts of things in it, bacteria, nematodes, those kind of things. Bacteria inhibit specific plant species. Some of those that she's developed and worked with have a good biocontrol match. And as the bacteria reduces the seed bank, plant diversity can increase. Here again, that's important. Why you need to have some type of perennial competition. You don't want to apply this to a 100% cheatgrass stand, because what you may end up with is worse than having 100% cheatgrass. So you need to have long-term management schemes. How are you going to manage the change? What's going to be there? How are you going to use it? So this is kind of a little bit of a repeat, but I guess the repeats are good to reinforce it in your mind. So we need to know that it colonizes the residue in the root, survives well at low temperatures, not competitive at warm temperatures, do not survive hot, dry summers. They produce plant suppressive compounds, and they're members of the genus Pseudomonas. Now, this was a previous presentation, and some of the bacteria isolates will actually suppress germination, which is a very interesting concept when you look at the vigor of the control group and what's going on with the other ones that are just barely growing. Again, this shows the situation with Downy brome, with the control. You see how vigorous it is, and that with the bacteria, the plants are weakened, not as vigorous. And here with the winter wheat, basically no effect. So we can know that Downy brome is being suppressed and wheat is not. These may be a little hard to see for you, but on this axis here we've got plants per square meter, and then the years. The blue is the control, and the cross-hatched is the actual result of the bacteria application. These plots are again scattered through central Washington over a period of years. Bacteria applied, the one at this time that she was using was called B7. So it's applied in 2009. You can see the reduction at this in this plot in the first year. The second year, the control plot is actually increased, and the other application spot has decreased. And again, on the third year, some of these weather plays a lot into the situations here. This particular plot, it had over 4,000 plants per square meter as a control, but the application area only had 1,500. That's really a great reduction. And then the second year, more reduction. And third year stayed about the same. And here at the bottom, you can see the effect of the first year. And if you think back to the one slide where there's probably about a 20% reduction in the first year, that's probably about this amount. And then here's some with cheatgrass and native range. Again in Washington, control plot here over 4,000. The applied area the first year was a little more than 2,000 plants per meter. The following year, below 1,000 cheatgrass plants. In the third year, just a few hundred. The bottom one, for whatever reason, everything worked just right in this one, and so by the third year, something happened here with the control, and in this year, even the control was a lot less, and the applied area was near zero. So the real project that we are involved with Dr. Kennedy on our CWMA is we had her on a tour throughout the county and looked at several plots, and she was just amazed that we had so much jointed goat grass. Just couldn't believe it. So we really looked for a place where we had some perennial grass for competition, because she wanted to put out some larger plots instead of doing the small plot work. So we selected a site that's northwest Louisiana in approximately a 10-inch precip zone, silt loam kind of soils. Soils that at one time been farmed and kind of dry farmed, not real successfully. It had been seeded to tall wheatgrass because the owner at that time was developing a livestock, or not a livestock, a hunting preserve, and he needed tall cover for pheasants and that kind of stuff. And, yeah. So this is, the bunches here are the tall wheatgrass in our area that we're going to use. The interspace is cheatgrass and China goat grass. This is actually in now in a livestock operation where it is grazed for a period of time in the spring. It's approximately 9 acres, so we're doing this on a plot that's more like a real life situation. We're hoping for similar results as previously. We were able to apply the bacteria last fall on the 12th of November. We'd had a fair amount of rain in October, and that helped us. It also hindered us on when we were going to be able to apply the isolate. But we ended up there in November on the 12th. It's, as previous slides mentioned, is soil moisture is very important, and temperature is important too. And so here we are on the 12th of November. You can't see the spray pattern here, but we're applying the product. That's Dr. Kennedy in the background. It was a cold morning. This is probably about 11 o'clock in the morning, and by 12 noon we had a snowstorm. So we were able to apply the product, get it on the ground, get the moisture to get it in so it wasn't being affected by UV light, and get it in there so it could start doing its thing. Hopefully, we've already looked at the plot a week ago. With wishful thinking, you think, oh, there's a difference between it and where it wasn't applied, but really we won't know if we've had any impact until next year at least. And hopefully by year 3 we'll know better whether this is going to work in this type of situation or not. So that is it. If there's any questions, if there is, make them easy.
Any questions, Mr. Tom? Okay, Tom, I'm going to ask you to repeat the question.
Okay.
Because this is microphone is where the recording the question, not mine.
Does weed suppressing bacteria only need to be applied one time, and it operates for several years?
Okay, the question was, does the weed suppressing bacteria need to be only applied one time, and how long is it effective? Basically, in the research that Dr. Kennedy has found that you have there's several things involved. If you apply it, if you think back to the one chart where by year 5 it had inhibited predominantly everything, but at the same time your population of the bacteria declines so that maybe by year 6 you don't have enough residual bacteria left to populate the roots to inhibit your annual grasses. So it's probably going to be a cyclic kind of application.
Any other questions?
Two questions. Did you see similar results with Medusa head? I didn't notice any data on Medusa head.
The question was about data on Medusa head. The isolates that Dr. Kennedy is using also works effectively on Medusa head, as well as cheatgrass and giant grass.
And then I think it wasn't wrong wondering how close are these to commercial production for large areas?
The question was, how close is this product to being able to be applied commercially? That's a really good question. She's been working with some different companies for a period of time to try to get this into the stream where it's available. Currently, they're working on trying to get the registration with EPA so that it is able to be applied. Right now everything is just on a trial basis. So the plot work that we actually did, the 9 acres, had to be approved by EPA and also Idaho Department of Ag. So, hopefully within a period of a couple, 3 years, I'd really like to see it available commercially somehow, because it's just another one of these tools in the tool bag that we all need to control a lot of these angry grasses that are impacting everything we do.
Other questions? Okay. Tom, could you back up? Recording.
Is she working on any other noxious weeds, Dalmatian toadflax, dogflower, any of those others with this same kind of technology?
Okay, the question was, is Dr. Kennedy working on any other noxious weeds? The ones listed or given were broadleaves. All her work has been on grassy weeds at this point in time, because early on, her research really wasn't on developing bacteria to control grassy weeds in rangeland. It was to control grassy weeds in wheat, because she's located at the ARS unit in Pullman, Washington, and it's a big wheat area, and the impact on grassy weeds in winter wheat can be really severe. So she'd like to get into some other avenues with some other broadleaves and those kind of things, but predominantly right now she's just working with grassy type weeds.
Any more questions?
I'm wondering if you know whether there are potential health effects in applying Pseudomonas. I've seen recently that Pseudomonas showing up on a list of 7 or 8 superbugs out there where we don't have antibiotics that would treat, or there's a very small number of antibiotics that would be able to fight, say, an infection caused by Pseudomonas.
Okay, the question was the effect of the Pseudomonas on health through application of the isolate. I can't give you a very good answer on that, because I believe that's part of what they go through in the registration process with EPA, and I'm just not very well versed in that. That would be a question you could maybe ask Dr. Kennedy, and if you need her email, I can give that to you too.
So the Pseudomonas fluorescens species is not a human pathogen. There may be other Pseudomonas species that are human pathogens, but not Pseudomonas fluorescens.
Thanks for your presentation. As you hike the rangelands in Washington County or down in the Hawaii, you see places where cheatgrass and medusa head is dying out naturally on an annual basis. And it's hard to contend about that. Are sites that be tested to see if the Pseudomonas is natural in the soil, causing that anti-looking basically die out cheatgrass?
Okay, well, the question was about in various areas in the Waihees, maybe populations of cheatgrass and medusa that dying out. Dr. Kennedy, I don't know how many sites that she has scattered through the Northwest and Idaho, Oregon, Nevada, Utah. Whenever she goes out, she's always digging in the ground and taking a sample home to look at art. And so I don't, I assume that she's searching for these other things that may be having other effects also. But I can't give you a really good answer on that one either. Sorry. I don't know how many isolates she really has, but she's got a whole bag full of them. And they all work a little different with different plants. So,
Any more questions? Okay, thank you, Tom.